The eye captures light and forms it into detailed spatial images, as well as transduces light into neural signals and processes them. The cornea and lens focus light, and when light reaches a different medium, it is refracted, which means light is bent to form an image on the retina. Lens shape is changed by the contraction of the ciliary muscles, which adjust the focus and forms images on the retina. This is called accommodation. The pupil, which is an opening in the iris, is responsible for the amount of light that enters the eye. Extraocular muscles consist of 3 muscles that extend to the orbit, which controls movement of the eye. The retina is where the first visual processes take place. It is the layered receptive surface at the back of the eye. Photoreceptors are located on the retina and they detect light. There are 2 kinds of photoreceptors. Rods, which are responsible for black, white, and gray vision, and cones, which are responsible for color vision. Different types of cones respond to different wavelengths. Both release neurotransmitters that control bipolar cell activity, and they connect to the ganglion cells, which form the optic nerve that carries information from the eye to the brain. Horizontal cells connect receptor cells to bipolar cells, while amacrine cells connect bipolar and ganglion cells. Rods, cones, bipolar cells, and horizontal cells do not produce action potentials, only graded potentials. Ganglion cells do produce action potentials. 2 functional systems that arise from rods and cones are a scotopic system, which uses the rods in dim light and gives this information to ganglion cells. It has a lot of convergence. And then photopic system, which uses cones in illuminated areas to differentiate wavelengths and facilitate color vision. These have less convergence. Rods and cones are stacked, so they increase the likelihood of light particles or photons that are captured. Since very few typically reach the retina, considering the extent to which the lens and fluid reflect the light in different directions. Photopigments capture photons. Rhodopsin is found in rods. Similar photopigments are used in cones. Rhodopsin has 2 parts, retinal and opsin. Retinal dissociates from opsin when light hits rhodopsin, revealing an enzymatic site. Here, phosphodiesterase breaks apart cyclic GMP, which holds sodium channels open. Light stimulation causes these channels to close, which causes a hyperpolarization potential. The more hyperpolarization potential, the less neurotransmitters that are released. It is important to remember that the visual system responds to changes in light, so either hyperpolarization or depolarization is sufficient. This cascade of processes leads to the amplification of weak stimuli, therefore increasing sensitivity, the slowing down of vision through the integration of the stimulus over time, and the adaptation to a wide range of light intensities. The eye is able to work over a wide range of intensities. The size of the pupil changes in order to accommodate for different light intensities. It constricts to admit less light through the influence of ganglion cells on information that reaches the superior colliculus. Range fractionation is when different receptors contribute to the handling of different intensities. Rods have low thresholds, while cones have higher thresholds. If receptors are not responding, sharpness of vision is impaired. The active receptors would be too spread out on the retina and cause a grainy or pixelated image. Receptors have a large range of adaptation. Photoreceptors adjust their sensitivities by shifting their range of response to match the ambient illumination. Varying concentrations of calcium ions that are maintained through the release of storage of these ions, therefore controlling photoreceptors' light sensitivity. When photoreceptor pigment is split apart, retinal and opsin slowly recombine, therefore determining how much photopigment is available to respond to light. The area you can see without moving your head is called a visual field. Visual acuity, or sharpness of vision, is best at the center of your vision and decreases closer to the periphery. The central region, or fovea, has a high concentration of cones, and optic discs are where blood vessels and ganglion so axons leave the eye near the nasal side of the fovea. A blind spot is not detectable because there are no photoreceptors in the optic discs. Rods and cones are more concentrated toward the periphery, leading to a decrease in visual acuity. Saccades are quick eye movements that bring visual information to the fovea to make up for the lack of visual acuity in the periphery. It's constantly occurring even when we think our eyes are still. There are no rods located in the fovea, but there are more in the periphery compared to cones. And finally, rods provide high sensitivity with low acuity, while cones provide high acuity with low sensitivity.